everybody, I'm Tall.
And I'm Small.
And today we are doing Khan Academy's MCAT Prep Biomolecules Carbohydrate Metabolism questions. Okay, so first question. How many total molecules of ATP are synthesized from ADP via glycolysis of a single molecule of glucose? So, how many total molecules of ATP? I know that in glycolysis you get a weird net issue that's different from the actual product.
Yes.
So there's two irreversible steps that use ATP and create ADP.
Yeah, there's three irreversible steps, but
Okay.
two of them in which you need to input two ATP. So you're investing two, and then you're creating four in total. So you net two, but because it asks for total production of ATP without thinking about what we had to invest to get there, we would come up with
4. So the answer choices are 4, 36, 2, and 38. From a single molecule of glucose, I know that we will produce 4. However, we're going to use 2 technically from our whole batch. So we would net 2, but we produce 4.
exactly.
And that's the right answer. Okay. Second question. What carbohydrates are reactants in glycolysis? Neither glucose or sucrose directly participate in glycolysis. Glucose and sucrose, sucrose only, or glucose only. Glycolysis. I think that sucrose is a disaccharide of glucose. So why wouldn't sucrose participate, right?
Yeah, except for our first enzyme, that hexokinase that phosphorylates glucose, is just looking for a bare glucose.
Okay, so you think that what they're asking here is they're saying really what is the actual starting point, even though a sucrose could just be broken in half.
Well, sucrose would be broken in half to glucose and fructose.
Oh, it's a glucose and a fructose.
Yeah.
Oh, okay.
Because you've got 9 chiral centers, you use a ketose as your second one, which is fructose.
Okay. So in this case, I would say glucose only.
Exactly.
And yep, that was right.
And it's literally lysis of glucose, glycolysis.
Okay, yeah. All right. Next question. In which organisms does glycolysis occur? Aerobic or only, anaerobic, neither aerobic or anaerobic, or both aerobic and anaerobic? Okay, so they would make me think that it would only be aerobic, but aerobic only matters for the electron transport chain. So, and if everyone's wondering why I keep asking Small these questions, because she is a metabolism expert, and I know nothing. So
that's being generous.
But MCAT's about reasoning your way through things, so it's all about just developing the right thinking. So oxygen has to do with the electron transport chain. Glycolysis provides some stuff that goes into the electron transport chain, like NADH and FADH2.
Yep.
Okay. So, but that doesn't mean that if you were an anaerobic organism and you were producing those things, they couldn't be used for something else.
Exactly. Well done. We know that if we go through glycolysis in the cytosol, we could either eventually, after Krebs cycle, producing a lot more electron-carrying elements that could help us with ETC. We could either go to ETC in that way, and then it would all end in oxygen, which would be aerobic, like you mentioned, or we can go all the way through glycolysis in the cytosol, and then that pyruvate production at the very end could be turned into something like lactate, or eventually, fermented into alcohol.
Okay.
In anaerobic organisms.
Okay. So I would say both aerobic and anaerobic.
Yep.
All right, and we got it. Also, I had no reason to believe otherwise. I guess that was the real crux of the thinking is, do I have a reason to believe why one of them wouldn't? Okay, question number 4. Which statement concerning the glycolytic and gluconeogenic pathways is correct? Both are anabolic, gluconeogenesis is catabolic, and glycolysis is anabolic. Both are catabolic, or gluconeogenesis is anabolic and glycolysis is catabolic. Okay, so anabolic means building up, catabolic means breaking down. Glycolytic means glycolysis. So glycolysis, you're breaking down sugars.
Yep.
Which means catabolic. Gluconeogenesis is creating extracting glucose from glycogen.
Not from glycogen.
I'm dumb.
Where we were essentially doing glycolysis in the exact opposite order. It takes us a couple more steps right at the beginning. So our last step of glycolysis, it takes us 2 steps to do that reverse process in gluconeogenesis. But yeah, essentially, and by necessity, they are the opposite. That they are not always, so like for glycolysis to happen, gluconeogenesis does not necessarily need to stop. There are always kind of a bunch of intermediates that we keep, and based on the environmental conditions of our cells, maybe, if we're running and so we're using up a lot of our glucose, maybe we need to do gluconeogenesis to build some more up.
So gluconeogenesis is creating glucose.
Yeah. So you're right.
Ah.
But we always keep a bunch of intermediates that can enter both pathways depending on what those cells need at that time.
Okay.
So they are the opposite. One is building up glucose and one is breaking them down. But we can change quickly between the pathways based on what our body needs.
Okay. Glucose, new, genesis to create.
Perfect.
Create new glucose. So that would be anabolic, and glycolysis would be catabolic, which is that answer.
Yeah, and cats like knock over crap all the time, right? And break things.
Oh my gosh, that's how I'm gonna remember it for forever.
And we've got a really sweet friend named Anna, and Anna likes to build people up and encourage them, and so that's how I remember it.
Oh, because Anna's a cat.
Yeah.
Oh no, she's Anna. Oh, anabolic. I wasn't even making the. I need to learn my metabolism.
She's a cat person though, so that's how I keep those are together in my mind.
Which is semantic memory. We will be going over that eventually.
Facts and information.
Okay, question number 5. What body conditions favor gluconeogenesis over glycolysis? Starvation, high blood sugar, increasing cellular levels of AMP, or low cellular levels of pyruvate? Okay, what favor creating glucose over using glucose?
Oh, I'm so glad that you said it like that. Well done.
So, creating glucose, we have a lot available. We have more than we need available. Therefore, we would use gluconeogenesis. So, if we had high blood sugar, maybe.
If you had high blood sugar, think about it in your Le Chatelier's principle. If I had high blood sugar, I have a ton of glucose, I would need to bring it into the cell and start breaking it down. I would need to take the glycolytic pathway.
Oh. So high blood sugar. Oh wait. Oh no, gluconeogenesis, we need to make new glucose. I keep thinking it's glycogen whatever.
Yeah.
Okay, it's not glycogen. Gluconeogenesis, it's because glue and gen are in there. So gluconeogenesis, creating, you need more energy. You have to create sugar from the junk you have lying around.
Right.
So starvation, maybe.
Yeah, that's what I said.
Okay. I would think starvation.
And so the other thing that you mentioned, another way we could probably introduce glucose into the system would be to start breaking up our glycogen stores in our liver.
Okay.
Yeah. So
which happens when you go into starvation mode, like ketosis.
Yeah.
Okay.
But our brain survives for a while off of those ketone bodies.
Right.
So,
and
yeah.
We'll check it, and it's right. Very cool. Okay, next question. In the redox reaction, pyruvate plus NADH plus hydrogen yields, in a reversible reaction, lactate and NAD+. Which reactant is oxidized, which is reduced? Lactate is reduced and pyruvate is oxidized. NADH is reduced and pyruvate is oxidized. Pyruvate is reduced and NADH is oxidized. Or lactate is reduced and NAD+ is oxidized. Oil rig.
Oil rig, yeah.
Or Leoger.
So, what I would expect them to ask more, if they ever gave you a question this easy on the MCAT, I think I would expect them to ask what is the oxidizing reactant and what is the reducing reactant, so that they're testing that. Do you still remember from Gen Chem that those definitions are kind of annoying to remember because they kind of mean the opposite of what gets done to those agents?
Yeah, it's a hard thing to fit in your mind, but good Lord, I would love to hear about the MCAT that didn't have an oxidation or reduction reaction question.
Yeah. So let me ask you this. If one of them got reduced in an oxidoreductase using, an enzyme that was doing that, would the other one have to be oxidized? What did I say first? Reduced?
Yeah. So if one gets reduced, does the other one have to get oxidized?
Yeah, that's my question.
Not necessarily.
Yes. Or it is not an oxidoreductase. So then we would change to one of our different classes of enzymes. So those enzyme definitions are hard to first learn, like the liases sometimes are annoying, but their definitions are set in stone.
Okay,
so they're really useful.
Oh, so you're talking about an oxidoreductase. Does one have to get oxidized? Oh, okay, yeah. All right, so in this case, I think pyruvate is getting reduced and NADH is getting oxidized.
Perfect.
So I could see NADH immediately. You have NADH, and then it goes to NAD plus. You're losing electrons. You're getting oxidized. But for pyruvate, adding a hydrogen to it, getting lactate, as far as reducing it, that doesn't like immediately make sense in my head, because I see pyruvate and lactate, and I don't see like a chemical formula.
Yeah.
But you're adding hydrogen.
Yeah, and the fact that they tell us, that it is a redox reaction means that, okay, they gave me that NADH is the one getting oxidized because I can physically see it.
Oh, yeah,
and so I must assume that the other one is getting reduced.
Okay, yep, and we got it right. All right, next question. What important reducing agent is an important product of the pentose phosphate pathway? There's a lot of P's in there. NADPH, NADH, NADP, or NAD+. So I do not know the pentose phosphate pathway at all. I would think, okay, pentose, that doesn't really help me when I'm looking at NAD, because I don't know what any of those mean. But phosphate, I think that's what the P is in the difference between NADH and NADPH. There's a phosphate in there. Am I on the right track?
Yeah, I don't know this pathway, so I couldn't tell you.
Okay.
All I could do was rule out 2 because it asked me for a reducing agent, and I knew that means it would get oxidized, so it has to be the reduced form. So I was between NADH and NADPH.
Ooh, so going by my stuff, it should be NADPH.
And I think you got it right.
Ooh.
And so NADPH is what the body uses when we're in an anabolic pathway. So even though I don't remember what the pathway is, when we go back and review it, I'm expecting it to be some sort of form of anabolic building.
Okay, yeah.
So let's see if it might tell us that in the hints.
All right. A reducing agent is an electron donor. Hydrogen atom contains 1 proton and 1 electron. NADH to NAD plus are not associated with the pentose phosphate pathway. NADPH is an important reducing agent produced by the pentose phosphate pathway. So that's kind of just knowing that pathway.
Oh, product of the pathway. Oh, anyway, yeah, we need to review it,
But the answer was NADPH.
Well done.
All right, so that's one of those pathways. Obviously, everyone knows you got to know the major metabolic pathways. At least know some basic steps of them. All right, next question. What is the best description for the energetics of glycolysis? Each step has a positive free energy change. Overall, under typical cellular conditions, glycolysis has a positive free energy change, though there are steps of both positive and negative free energy change. Overall, blah, 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 basically the opposite. Sorry, it's a long answer. Each step of glycolysis has a negative free energy change. I am going to say, I saw that you clicked something. I didn't see what you clicked.
Okay.
So I think overall it has to have a negative free energy change. Because you're producing more ATP than you're consuming.
Well done.
Although there are steps of positive and negative free energy along the way.
Exactly.
Because you have some of those reversible ones that require an input of energy. Yay!
Baby claps, baby claps.
We wanted to clap, but it doesn't sound good coming through the microphone, so I got a silent clap.
It makes me excited. Really quickly, I do want to mention, I said in the question previously that NADPH is used for anabolic pathways, but I do want to be very careful in saying that because in gluconeogenesis, which is the first anabolic pathway that I think of, NADPH is not used. NADH is used. And that is because in its equivalent step in glycolysis, NADH is what we use for that one as well. And so since it's freely lying around in the same areas in which this pathway is also happening, we just use NADH because it's simpler than pulling in a different molecule.
Okay. Very cool. Very cool. All right. Question number 9. The in vitro conversion of glucose to glucose 6-phosphate, which is, then they give us the thing, glucose plus inorganic phosphate, reversible action with glucose 6-phosphate and water, is highly endergonic, though in the setting of glycolysis proceeds in a spontaneous and irreversible fashion. How is this achieved? Okay, so they tell us it's highly endergonic, meaning that there is a positive delta G. However, when you're in the setting of glycolysis, putting a phosphate on a glucose is spontaneous and irreversible. So, how is this achieved? Through coupled ADP condensation reactions, through coupled ATP hydrolysis reactions, high concentrations of H2O shift equilibrium to favor G6 phosphate formation, or endergonic reactions are always spontaneous and irreversible. Okay, that's wrong.
Yep. Well done.
How is this achieved? So you're producing water. ADP condensation reactions. They want to know putting a phosphate on a glucose. ATP hydrolysis reactions, high concentrations of H2O shift equilibrium. I mean, that makes sense Le Chatelier-wise, but I feel like there's high concentrations of water everywhere in the cell, so I wouldn't necessarily pick that. That might like break Le Chatelier's rule or something because we got water everywhere. Or through coupled ATP hydrolysis reactions. Coupled ATP hydrolysis. I think I remember the formation of glucose 6-phosphate to be irreversible, and meaning that maybe it requires an input of ATP. So I'm gonna say through coupled ATP hydrolysis reactions.
And you are right. So this question is one of my favorite questions because it sounds like a whole bunch of word garbage that's testing you on something that might seem harder than it actually is. And it's just asking you that if we have something that seems like it's a really endergonic reaction and it's an important part of metabolism, how do we get that going? Yeah. And the answer is almost always we'll couple it to breaking up ATP because that is so exergonic.
Yeah.
And you know, so that's one of the basic tenets of metabolism, that we couple endergonic and exergonic reactions and get an overall negative delta G.
Okay, cool. I like that. That was a good question.
Yeah.
All right, and we got it right. Okay, last question. Which glycolytic reaction cannot be directly reversed in gluconeogenesis? Is it 1,3-bisphosphoglycerate yields 3-phosphoglycerate? Glucose yields glucose 6-phosphate, glucose 6-phosphate yields fructose 6-phosphate, or glyceraldehyde 3-phosphate yields 1,3-bisphosphoglycerate. So which glycolytic reaction cannot be directly reversed in gluconeogenesis? So we're not talking about glycolysis here. However, it is a glycolytic reaction. We want to know gluconeogenesis. Well, didn't we just say that glucose to glucose 6-phosphate is irreversible? However, I don't think that occurs in gluconeogenesis. Conversion of glucose 6-phosphate to fructose 6-phosphate, I feel like that also does not occur in gluconeogenesis. If we're trying to create a new glucose, we're not taking a glucose and then creating a fructose from it.
So they've written this in the glycolytic layout, in which they're telling us what we would start with in glycolysis and then the product of that reaction. So reverse it because we're talking about gluconeogenesis.
Oh.
So don't rule them out that way.
Oh, okay. Well, let's start it over then. Okay, so glucose 6-phosphate going to glucose, I feel like that could be reversed. Maybe not. Wait. Okay, let me look through the rest of them. Fructose 6-phosphate to glucose 6-phosphate. I feel like that can be reversed.
Yep, isomerase. Pretty solid bet.
Okay, 1,3-bisphosphoglycerate going to glyceraldehyde 3-phosphate. Phosphoglycerate, maybe? But once again, you're putting a phosphate on. I feel like that tends to want to be irreversible. And then going from 3-phosphoglycerate to 1,3-bisphosphoglycerate. Oof, I am really out of my element here.
So this is where I'm literally looking at molecules in my head and remembering that steps 1, 7, and 10 are irreversible and have their own enzymes that are opposite of the glycolytic ones to do these steps. So 1, 7, and 10 actually has 2 enzymes that do step 10 in gluconeogenesis, which is the first step of that pathway.
Okay.
So the rest of those, I'm thinking in my head, well, could that be reversible? And the answer really is yes to all of these, even though step 6 of glycolysis is actually a redox reaction, it is reversible. And so only 1, 7, and 10 would need, are not directly reversible. So the answer really is glucose to glucose 6 phosphate.
Okay.
So you would directly need a phosphatase to cut it off.
Ah, okay. Yep, and that was right. All right. So, yeah, lesson learned today is just go through those main pathways and memorize. I mean, if you can memorize all the steps, that's only going to help you more and more. But I think at the very least, you just have to know about input, output, about reversible parts, irreversible parts, and have a basic understanding as how it affects the entire metabolism and what conditions you might see certain things.
Yeah, exactly. And especially those overall themes like coupling something that's highly endergonic with some things that might be exergonic, and how those intermediates play a role in being quickly able to change directions from creating glucose to using glucose. And I need to look up what the pentose phosphate pathway is.
Don't we all? Strong work, gang.